And so what do we have to do? What do we have to do to increase the impact and usefulness of the system? So I guess I'm sorry we don't have more people, but let's go for it. King, so do you want to start off and just talk about the paper, and then I'll show the portal more, or you can show any part of it you want, and then I'll pick up and.
Yeah, sure. You can like interrupt me at any time when you find you can add more content, and I will just briefly talk about what we have in ToppCell right now and how we use it for exploring the COVID-19 immune system. So I will share my screen, I guess. Can you see the screen, the PowerPoint?
Perfect.
Yes.
Thanks. Go to begin. So thanks everyone, and I'm Kang, the PhD student in Dr. Bruce Arono's lab, and today I'm going to talk a little bit about how I can use ToppCell to explore the host responses in COVID-19 infection. And here is a recent paper that we just published about this tool, the web portal, and how it can be used to explore something we are interested in COVID-19 host responses. And as I mentioned several times in maybe in other presentation, one of the highlights that I think in ToppCell is that it can handle super complicated biological metadata in the single cell data, which is gaining more and more popularity. And with more and more data generated, more people care about different experimental design instead of just doing the cell atlas in the healthy donor. For example, they look into the perturbation and the cells in different compartment or tissue, as well as different developmental stages. And for the cell itself, we can use unsupervised clustering or like reference mapping to get more detailed knowledge about their those cell components in a specific data set, including lineage, cell class and the subclass, as well as spatial information and other cell annotations mapped from the reference data set. And we also sometimes care about the effect of donor as well as sequencing platform and other batch effect. Sometimes we want to remove that, but in other cases, we also want to look into that. And with those complicated metadata, we were thinking, can we handle it in a better way that people can understand it in a systemic way? And this is an example of annotation that we have in a very normal COVID-19 dataset, which includes like the disease condition like healthy and COVID-19, as well as lineage, lymphoid and myeloid cells and the cell class. And with those levels of metadata, we can use ToppCell to analyze the signatures in each level. And this is another example that we apply ToppCell into. Here is an example of a single cell dataset for hepatoblastoma, and we got 11 subclusters in hepatoblastoma tumor cells. And with that, we just divided the metadata of cell types into 3 levels. One is cell class, and the other is cell sample group, including background, human tumor, and xenograft tumor in mouse. And we visualize the tumor subcluster signatures in each sample group and see if they are consistent or heterogeneous. And with that, we can do it in both ToppCell and ToppGene. ToppGene can be used for gene enrichment analysis. And when we combine all those signatures, gene signatures from different clusters, we can actually put it into ToppCluster for building a functional map, and then further making more complicated network to extract the functional associations of tumor-related biology and their association with our genes derived from the tumor cluster signatures on the ToppCell. And just from here, we can get several highlights that this system, this eco environment can help us look at the global gene signature and create functional map, as well as creating pretty complicated network. This is a web portal that we built for ToppCell, and currently we have already curated more than 70 separate single cell datasets. They are all they are all after quality control and well annotated. And with that, we combine with top gene and the top cluster, which consist of the top toolkit, and each of them have separate functions for either like gene module generation or gene enrichment analysis and the comparative functional analysis. And this is a example from our paper, and we just do meta-analysis for combining several COVID-19 datasets, and from the columns, they are the metadata of cells, and you can see this is pretty complicated. Basically, we have conditions which is either control or severe. Those are the cell sources, and they can come from control donors or severe patients. And we also annotate their locations, which means the source of the tissue source of those cells, including bronchial alveolar lavage, lung parenchyma, and the PBNC. And we have two other levels of cell group and the cell class, which are more detailed cell type annotation. And with that, we just build a ToppCell task and then conduct differential analysis for each bin here, and then derive a top 50 differentially expressed genes in each chunk. And you can see they are the annotation for those gene signatures. They have lineage, condition, cell group, cell class, and the location annotation for those genes. And each gene module or gene signature list contains 50 differentially expressed genes. And to better visualize, I just labeled them on the left, where you can see different lineage of cells, and the C means control, the gene signature derived from control cells, and S means the gene signatures derived from severe patient. And we just align them side by side to have a better comparison between the gene modules in control patient and the gene module, the corresponding gene modules in severe patient. And here I just want to highlight some part. For example, this part, actually they are the signatures derived from the control myeloid cells. You can see the blue box on the left and on the right. And the left part shows those gene modules derived from control cells and how they look like in the cells in control, and how those corresponding signatures derived from control looks like in the severe patient. And vice versa, we also look into the gene signatures derived from myeloid cells in severe patient here. And you can see they are pretty bright on the diagonal pattern. And when you look at those signatures, specific signatures derived from severe patient, and you can see how it looks like in control. And some of them are still preserved, for example, this part, but some of them are less bright, which means their expression may be downregulated. And because we just compressed them pretty a lot here, and each small chunk contains tens of genes, and this is just a global view to pinpoint which part we can focus. For example, this part, if the signature on the control and on severe, they are very different. We can just focus on this chunk of gene and zoom in to see how exactly those detailed genes looks like differentially expressed, and we can compare between the control and the severe patients for the myeloid signatures. This is just an example of very compressed, how we can use it in a compressed way, and how we can look from a global view. And After that, I want to briefly discuss the interface of ToppCell because we just enable all the interaction in the web portal, and the users can play with our current more than 70 single cell data. And here's just an example of one of the PBNC COVID-19 single cell data and how it looks like, including the gene signatures on the right sidebar and different element on the web portal. And right now we have different projects which covers several single cell data sets in each tissue or in each species. And welcome to explore more detail about that in the using the link on the bottom. And here for each interesting gene module, which are we use default 200 differentially expressed genes for the definition of each gene module. And for each gene module, you can click it and send it to top cluster or top gene for enrichment. And then in the top cluster, you can either use functional enrichment, which you can get side-by-side comparison of different functional enrichment for different gene lists. Or you can also click the interaction network to see the gene interaction network and or protein interaction network. And to enable the the standalone version of ToppCell in local computer, we also build ToppCell in Python. And this is the link where you can just use Python and ScanPy to automatically generate all those gene modules. And with that, we just built a signature atlas for COVID-19 immune cells, and then we explore different biological process that happens in mild and severe patients. And there are several highlights that we found using meta-analysis of 8 single-cell dataset for COVID-19. And they are the link for each tissue, the atlas of single-cell data in each tissue, including PBNC, bronchial alveolar lavage, upper airway, lung atlas, and other tissues. And they are the meta-atlas, which we just do meta-analysis and combine several interesting data set together and extract the cell types that we are interested in. For example, T cells or platelet, and build new atlas in ToppCell. And combined with other immune-mediated disease, we can better understand how those immune changes look like in COVID compared to other immune-mediated disease. And this is an overview in our study, like how we use those 8 public COVID-19 dataset to do our meta-analysis. And yeah, this is our diagram of the analysis pipeline. And to better, because previously I just show a global view of signatures. Here I want to show a more specific zoom in view for specific cell type. For example, we know that it was widely reported that neutrophil and the macrophages were attributed to the cytokine storm in severe COVID-19 patients, which can induce very severe symptoms. And we just extract those 2 cell types. One, you can see in the panel A, there are 5 subclusters of neutrophils that we identified after combining 8 single-cell dataset. And on the bottom, we highlight the macrophages because macrophages only exist in lung as far as we know. They are not in the PBNC. So we just highlight the subclusters of both tissue-resident macrophages and the monocyte-derived macrophages on the UMAP of lung. And they are specific subclusters. And for those, we can also use ToppCell to derive their signatures. Here on panel C, we just derived the top differentially expressed gene modules for those 5 subtypes of neutrophils. And you can see actually the neutrophil 0 and 1, they are pretty similar with a lot of overlapping genes on their gene modules. However, for neutrophil 3, actually, their signature pattern is pretty distinct. You can see on this pattern that highlighted in neutrophil 3, we can see genes that only upregulated only strongly upregulated in neutrophil 3. And when we do gene enrichment analysis, we find neutrophil 3 is actually a hyper-inflammatory neutrophil. And when we look at the source, actually, it's mainly coming from the bowel, bronchial alveolar lavage, and others were mainly coming from PBNC. And with that, we can tell that actually the neutrophils that appear in the lung or bronchial alveolar lavage is more inflammatory than the neutrophils that we found in PBNC, although they are all coming from severe patient, but their signature are pretty different. And we can also, sorry, this view is a little, there are too much information, but the basic point is that we can just, so on the rows, we select the immune-related pathway or inflammatory related pathway, and they are in different categories. And we can show the enrichment score using top cluster for all those subtypes of neutrophils, as well as subtypes of macrophages. And we can compare side by side. For example, for those cytokine-mediated signaling pathway, how they look differently in different subtypes of neutrophils and the macrophages. And the one highlight is actually compare with the tissue-resident macrophages. Those monocyte-derived macrophages has very significant upregulation of inflammatory pathways or cytokine-mediated pathways. And there are a lot of details here, and we have more detailed discussion in our paper, and are not going to too much details in this presentation. But yeah, another view is that actually we can also look at another very important cell type, which is platelet, because coagulation is widely observed in severe patients, which can lead to very severe symptoms. And we just extract the platelet from 8 dataset and do the subclustering for the combined platelet cells. And we can see actually there are several subtypes like platelet 0, platelet 5, and platelet 2 and 4. They are pretty specific to severe patient cells. And when we do that, when we do their when we do the ToppCell like differential gene visualization, we can highlight several genes that are more specific to the subclusters in severe patient, as well as some patterns that are specific to both mild and severe patient. And it involves in actually a lot of manual work. And we just like put forward a panel of genes that could be highly associated with the coagulation or immune thrombosis in COVID-19 patient. And with those genes, we use top cluster and cytoscape to build a network which can potentially explain why in some of severe patients, the coagulation becomes so bad. And this is the network that we associate like different panel of genes, including pan-platelet genes, the genes specific in both mild and severe COVID-19 patient, as well as the genes that are only specific in severe COVID-19 patient. And we highlighted the association of those genes with coagulation and the thrombosis related pathway and show on the surrounding box. And we assume that those genes that we highlighted using a sicked lines could be potentially attributed to the very severe symptom of coagulation in the severe patients. And I think I will stop here with that. I want to acknowledge all those people that contribute to this project. And the ToppCell is still in development, and there's still a lot of drawbacks that we need to improve. So we are welcome to all those suggestions and the feedbacks for our tool. And Bruce, you can take over, I think.
Sorry about that. Okay, that was awesome, Kang. I'm going to show from this other computer. And so let's get into this. So, okay, so here's the general interface to ToppCell. And you can you can search across the whole database for cells or genes, um, or you could go into a particular project group that has data sets that are grouped together according to some theme. And so in this case, in the COVID-19 atlas, here King's beautifully summarized a bunch of different data sets and the meta-analysis of them according to which compartment they were sampled in, one or more compartments, um, and then what the states are, the disease states are. And so you can now do a scan for some gene, let's say that you're really interested in. King was talking about those inflammatory um, neutrophils. So let's say we look for, one of the markers of that neutrophil 3 is like IFEDM2. So, or you can look for any gene. And when you search for a gene in the signature atlas that's generated by ToppCell, um, it it says, okay, look, you search for IFID-M2, here's the and everything is just within the COVID-19 atlas, and then here's the data set that where a signature was drawn from, and you can sort by any one of these things that's across in the columns, thanks to Eric's amazing engineering. And so what you see then is the rank of the gene within the signature. Here, this is, and the signature is it's a gene module. In this case, it's neutrophils that were pulled from saliva of patients that were very severe and sampled within the time window of 28 to 40 days after they got COVID. And this is the mean log2 TPM expression of that gene in the signature. And then here's a statistical score for how significant it was in that cell type compared to other cell types. And you can see even in like COVID-19 versus the world, the stat test for that to get a signature, it's still an incredibly significant marker of COVID-19 response. And so that's cool. And so you could follow any of these links. And so what can you see in that? Here, if you hit this heat map view, it pops up a heat map of that view, and then there's a couple of things that are in one of these heat maps, and it's like King was saying about the metadata. Is we try and get all the metadata to help you understand the context of this. So the browser's trying to get back memory. Okay, so in this case, there's a couple of compartments, bronchoalveolar lavage and pulmonary fluid. There's PBMCs, peripheral blood mononuclear cells, and then saliva that were sampled in here. And you've got different severity states. These are severe patients in the 12 to 22 period that were not treated with steroids. And so let's actually sort by that. So here's this. Okay, there's a bunch of different treatment groups, and mild, moderate, different time intervals. Let's just concentrate on. Well, I'm going to do corticosteroids. And so let's just look at they had a beautiful set of samples in here. This is Ren et al, isn't it? Actually, I forgot to look. I think so. Yeah, cortico. It's not. I'm sorry. All right, let's just let me just do this. We'll do the simple test of just looking at the signature. So we'll arrange by lineage, cell class, cell type, and then cell subtype. And here's the signature that we selected for down here. It's neutrophils in that this time interval of that patient group. And so when you see, okay, this is neutrophils, it's a there's its cell ontology entry, and then here's these neutrophil subtypes. And this, we didn't go to any of the subtypes, so this is a signature that's just present in all of the neutrophils of the severe individuals. So, and let's just do a sanity check because we searched for iFit, wasn't it? Oops. You have to search in rows and iFit. And so here's all the iFit genes in there, and you can see there's iFit M3, M2, M1, and as you go up this lineage, so myeloid cell neutrophils are part of granulocytic myeloid cells, and they're just generally part of myeloid cells, and then this was the group to comparison of severe um, um, versus non-severe of day 28, 40 in saliva. So these are trying to understand differentially expressed genes in saliva. And as you get more and more specific down here. So let's see, let's just grab this last signature in here, and then you can select the genes in that list and then go explore them. And that's kind of what we preview that in that the view of the data set. Let me just explain what that means. And then these are looking for the enriched features of it. And so now going back here to, instead of going to the heat map view, you could have chosen to go to the view of the data set, and then you could start to browse into it and see what's going on. Okay, so here's the data set Ren et al. That I was looking for. And then this is a preview of the data set, and we're talking about the saliva and in severes at this critical period way into it. And we were looking for in the myeloid cells, in the granulocytic cells, the neutrophils, and then there are these neutrophil subclasses in here. And in this case, we just looked at the pan neutrophil signature, and this is that. So I click that gene list, and then here's that list of genes, and this gives you a preview of the functions that are present in there. So incredible p-values for related to activation of immunity. And so neutrophils have both a direct role in as defense cells, and then they also signal to other cells to say there's something very bad going on here. Come into and help me fight off this infection. So, and then that gets into this interactomic analyses that King was talking about. So let's say we just do incredibly simple test of neutrophils to T cells and B cells. And so we can ask maybe two different B cell types, let's say CD4s and okay, so we'll just do CD4s, CD8s, and then so now I've got three signatures in here, and I'm asking the question when I set it up like this: is there communication between the neutrophils and the T cells? And you can limit that to ligands and receptors by clicking those two boxes. Then you get this preview of how many genes qualify for ligands in any of those three signatures, and it's 11, 6, and 4 genes in the neutrophils and T cells, two different mature T cells. Or you could go to receptors and see that it's a huge number of receptors in those three cell types. So let's just look at the communication between ligands and receptors in those three cell types, and then you could click that button there, enrich, and then you have this option here, interaction network is the option to look at, and then all it does is look for the protein-protein interactions against the database of all protein interactions between those three cell types. And so now you get this initial primitive view of the network here without any other information other than the protein-protein interactions, but you choose to export, and then it downloads the XGMML, and you can open that from Cytoscape and get some of those beautiful views that King was showing in the network analysis. So there's that angle of looking for the cell-cell interaction. There's the angle of what's the actually how do these genes work together in this host defense function um, from the neutrophil side, or forming an intercellular network where you get a cascade of activation of cells that are then working as a team to fight the infection, and what are the consequences of that, and then how does that differ between individuals that only had a mild degree of infection, or ones that are individuals that became critically sick and either died or needed ventilation, that sort of thing. Um, okay, so that's one view and one set of functions, and it keeps going and in really interesting ways. I wanted to show um, then a summarized view. You can put all this information together, and one thing that Kang was showing in that overview of all the cell signatures in these different conditions. So this is after maybe 20 minutes of work. You can get a view like this from those data that we were showing. And in this case, I've just zeroed in on the day 12. I don't know if you can read this in the cursor. Is that too small? It's pretty small. Um, but down here, it's the red group. Red group of columns are individuals um, bronch, um, bronchoalveolar lavage fluid, and it was with no steroids in that 12 to 22 period for individuals who were severely sick. And then this is also individuals who were severely sick who went on steroids that got dexamethasone. And dexamethasone had a huge effect. And we now know that dexamethasone is one of the most successful drugs to keep people from spiraling out into a critically ill condition where the lung ends up getting absolutely destroyed. And so it does something to break this signaling cascade of cells that's leading to this inflammatory crisis, and you necrose off lots of different cells. So okay, so look down here. These are genes. So this is 10,000 genes from top to bottom. The genes that are signatures of each of these cell types. So here's epithelial cells, lymphocytic cells, myeloid cells, and granulocytic cells, and then the subtypes of each of those cells, monocytes and CD8 T cells, subtypes of the T cells, and then this is a post hoc analysis here. So here's steroid minus untreated, and so then here's a bar graph plotting the difference between the steroid the value of the gene's maximal expression in the no steroids condition and the versus the with steroids condition. In this case, these are all minus numbers here because the genes are all in this block here. They're all strongly downregulated by having been treated with steroids. And then over here are genes that are all upregulated by steroids in the corresponding cell types as you go left to right. Whereas down here in blue are all the genes that are similarly expressed independent of the steroid treatment or not. And so then the next step is to unravel what are these genes up to. So let's say there's two angles to consider, at least two angles to consider. One is what's the number of cells that are present in each of those samples and sample groups, and then the other is what are those differentially expressed genes that are like indicative of severe, non-severe, treated with steroids, not treated with steroids, early in the disease versus late in the disease. All of those are critical metadata parameters that are going to inform you about the biological processes that are going on here that are leading to either a successful defense or an unsuccessful defense against the virus. And so you can, in the top is a count of the number of cells in each of those classes. And we're still fiddling with a little bit better way to depict that. But and the principal problem is normalizing the cells between different sample groups so that we can see the proportional changes. and anyway, we're here's the raw data, and then that about the counts of those cells and those types, and it's going to look even better once we normalize it. But anyway, so let's say so there's a couple of things to consider. Look at all of these cells now that are coming up as being very abundant in the steroid treated versus the steroid untreated, and then what are all these genes that are turned on? What are all these genes that are turned off? And there's something different going on between the myeloid and the lymphoid cells as well. Something very different. So just to unravel it a tiny bit, I mean, this is an amazing thing. So here's lymphocytes and myeloid cells, and they're the genes that are strongly turned off by steroid treatment. From there to there. So there are all these genes that are well suppressed, incredibly well suppressed. And so if you just grab those genes here and then just do a simple enrichment for them, this is what they show. And it's talking about adaptive immunity and inflammatory cytokine, tons of, so this is kind of inflammation 101 and steroid treatment 101, things that we've known for 20 years that steroids can suppress the cytokine storm. But then if you look at the underlying cis-regulatory elements in those genes, look at that. It's all NF-kappa B. Incredible enrichments for NF-kappa B in those, and then when you take what these genes are and then you spread them out over those different cell types, you'll see it's NF-kappa B pathways in those different cell types. Because remember, we were looking at this view. These are all independent cell type specific signatures that are stopped and are really turned off by the treatment with steroids. And whereas these genes here are in the exact same lineages, and these are all the genes that are turned on by steroids. And there's something else going on here. These are proliferative cells here too. So that's telling you now, oh, I'm getting some really good. I'm reinstating proliferation I'm getting like a ton of these CD4 cells that are proliferating under steroid treatment. And this is something that is not well appreciated by steroid treatment. The NF-kappa B pathway suppression, that's extremely well understood by, and there's hundreds of papers that go into that, over the past 20 years. What's not understood is that in context of this disease, that now you restore proliferation of T cells by the treatment with steroids. It's a little counterintuitive because there's some T cells that are thought to be killed off by dexamethasone treatment, but under this circumstance, no, it's very clear that you're restoring proliferative potential to those cells, and the extent this is high resolution view. This is what we'd call like the fourth generation of single cell profile data in COVID patients. This is just a brand new release data set just in that past few months from this fabulous group in China, and Ren et al. And so this is, and they, it's not analyzed in the paper at this level at all, but this is sort of what we think can get done within the ToppCell environment. Is look at that. Just one more look at what these genes are that are activated in the steroid treatment. The opposite of the NF-kappa B. Same exercise, and you actually I already did it over here, and genes that are very critical for immunity. And so it's the acquisition of intelligent acquired immunity, where you really have to learn about adaptive immunity to have antigen-specific immune responses. So that's all of that is restored in the steroid treatment approach. So okay, so that's probably enough of blah blah blah. And if there's, we'd love to hear just any questions or discussion, anything that could clarify just what could make it a more likely event for you to get onto the tool and use it and try and extract useful information.
Yeah, it's not necessarily like something that will make me more use it, but I think it will be useful to see other types of data integrated too. So I know that there are proteomics data sets out there. We will have one soon. It's not out yet, and it's not for me to share. So when it will be there, I'll let you know. But
Okay.
are you planning to do that, or?
Oh yeah, absolutely. I've been working on the proteomics data actually, and I can show you what the view is that it's going to be on the system. But that I really want to see um, what um, what proteomic signal by transcriptomic signal is and really in specific ways. And so as you guys get more and more,
I know we have already bronchoalveolar, or well, no, it's actually lung exudate that comes from the intubation. And we will have also different cell types, sorted cell types, proteomics, and lipidomics from donors with and without COVID.
so here, this is this dissection of the proteomics data. That here's all the immune cell signature. Here's the mesenchymal cell signature. So let's say we take the immune cell, or I'm sorry, mesenchymal cell signature, and then you can see how does it, what's the overlap in the other kind of authoritative signatures that we get from the single cell data set. So yeah, this will be pre-processed and they're able to preview. So this is 326 genes is very good depth into mesenchymal cell signatures. So that's pretty exciting just on its own. And that's a false hit. And so now just going against the, you can limit the comparison. So we'll just go against a couple of different lung data sets. So human lung cell atlas and weighing it all. Okay, so that's 3 good. There's one more. And so now basically you'll see the overlaps to the ToppCell signatures, and then you can go right to those heat maps and then put on, just paste on the new columns for that proteomic data. So down here.
What paper was that, Bruce?
That was the sorted human cells proteomics data that you guys released.
Oh, yeah, I was more wondering, can you interrogate proteomics data set from COVID?
No, I don't have any. Do you have any?
Yeah, that's what I was saying. We will have some, but there are some online.
Yeah.
But there are some online that could, be leveraged already.
Oh, fantastic. Yeah. Yes, but even Luminex as well as actual mass spec-based proteomics. I would love that actually if you could point it. But Jeremy, look at this. Here's this overlap now, and so we're getting mesenchymal, and so something that comes out. It looks to me like when I look at these cells, it's the hit is much more significant for mesenchymal smooth muscle cells. So I think that those fibroblasts were very enriched for smooth muscle cells and myofibroblasts rather than the matrix fibroblasts.
Yeah, I agree.
Yeah, and so you can unravel that here and get, so if you in this case you can go let's get some of these, let's get a smooth muscle subtype. It doesn't go very deep.
But just to come back to the COVID question, I think like looking at whatever like lung exudate coming from the patient and see for what's the cellular origin of those proteins that are expressed in the biofluid may help understand what's maybe driving disease too. So
yeah, and there's also a very significant number of proteins that are not well measured by the, like interleukin 6, that are not measured by single cell data sets. And so it's too critical that their levels, they can't go to too high of a level, and they need to turn over fast, so the mRNAs are made non-polyadenylated so they don't get labeled in the single cell data set. So there's a distinct dark matter problem of transcriptome that the proteomics can resolve, and some of those can be functionally incredibly important proteins. Okay, so here's the smooth muscle thing. All you do is you just go back to this proteomics data, pull out what are these genes in here, and then you just basically paste those genes into the transcriptome data from the proteomics, and then you stipulate that it has to be an exact match, and then see where we're at on those. So here are all those genes that were a hit in the proteomics, and when you look over this, it's smooth muscle and smooth muscle. So you can see that it's two smooth muscle cells that are deeply enriched by those hits. And yeah. But I think we want to be able to set up XY plots so that we can really see like outliers, protein versus transcriptome.
Hi, Bruce. This is quite comprehensive tools. And I have a question up here, like a cell communication tool. You said it's based on the PPI?
Yes. Yeah, that's
not based. If it's not expressed in the same cell, the PPI will not happen, right? So you actually check the expression first?
It's all dependent. You're only doing this for genes which passed a threshold of expression in the cell type.
Oh, okay. So not
so
really.
The only inputs are like expressed in cell type A, expressed in cell type B. Of those, who's the PPI, and you can further restrict it to those that have PPI of who are a ligand, who are a receptor, or just any cell surface gene, not necessarily a signaling receptor.
Yeah. So I assuming like between cell and a B is based on the ligand and the receptor, right? So and the inside the cell you can see the PPI, right? You cannot. Both.
Yeah. So exactly what's a legitimate intercellular signaling molecule? Not that many. So it's genes that are ligands that are definitely released from the cell, or extracellular matrix components that are secreted that then the cell can interact with from one of its surface receptors, or and then after that, it's the PPIs within the cell are very important to understand the networks within the cell.
And in terms of your ranking of the candidate gene, what is the what mostly based on? Because you have so comprehensive metadata. I assuming you have based on different metadata, you have a different ranking system, right?
Yeah. So here's 3 different significance scores about a gene within one of these modules, and so this is just its log to TPM expression value. That's just after normalization of per million counts, and then here's its statistical score for let's go for its how specific it is. So these are going to be really good biomarkers of a given cell type. If it has a good T-score, it'll be really specific for whatever the cell type is that it's coming out of.
Yeah. So, for example, if you choose the severity or the response to the steroid treatment, it will be different ranking.
Absolutely. Yes. Absolutely. Yeah. So our first level ranking is just based on for that, whatever the name of this module is, compared to its its comparison group. So in this case, airway smooth muscle subclass 2 as compared to medial lung cells.
And you probably did some comparison with the original paper, like for example, the severity markers, and show the association of the prediction.
Yeah. Yeah, that generally is that's pretty that's very straightforward. Usually we do way better than what's in the paper.
Oh, okay.
Almost always.
That's great here. Thank you. Thank you both.
Yeah.
Yeah, one thing I want to add is that actually our statistics is pretty limited currently. We only use t-test, frankly speaking. And actually in Threads or in other like newly published data methods, they have more advanced statistics which are better, like modeling the single cell data for their negative binomial distribution property. And with that, it's always better to use more advanced algorithm to really get the differentially expressed genes. Based as far as we know, like t-test is not that problematic, but still a lot of to improve here. And the scores that Bruce just showed, including the mean and the score, they are the t-test statistical output. And for example, the current state of art could be masked, but we are still trying to include that in the following versions, which can help us better understand more like more generic markers in single cell data. And because currently we are still observing a lot of like impact of those ribosomal genes or other like genes may be related with batch effect. But in the next version, we hope to get that better adjust. And if you can give more suggestions, that would be great.
Got you. Thank you.
All right. Thanks, everybody. If any comments, please send our way that could help improve the really getting the impact out for using the system. Well, thanks.
Hi, everyone. Move to another zone.
Oh, no.